The next presentation is construction of Bionic Nanoparticle-Loaded CLU protein peptide targeted delivery system and its role in treatment of intestinal radiation injury by Zhang Keyuan from Naval Medical University.
Good morning, respected judges and dear students. My name is Zhang Keyuan, and I'm from the Naval Medical University of China. And our topic is construction of Bionic Nanoparticle-Loaded crude protein peptide targeted delivery system and its role in the treatment of intestinal radiation injury. Sorry, I need to get. In recent years, the nuclear radiation technology is widely used in the national defense and many other fields. And with the rapid development of the Chinese Navy, the nuclear power technology is getting better and better too. But at the same time, there's a growing risk of acute radiation injury to the ship crews. And as we all know that, the doctors, especially for the radiologists, are under the risk of the radiation injury too. And for the patients who get in the radiotherapy, for the cancers are also. And their safety are threatened by the accidental radiation injury. And researchers have shown that the intestine is the most vulnerable organ after the radiation injury, with epithelial cells necrosis and the stem cells damaged. And the proliferation and the differentiation of the residual stem cells are the only hope for the regeneration. And researchers have also shown that after the radiation injury, the CLU gene expression rises significantly. And the CLU-traced mice model shows that the CLU-positive cells will replace the ALGR5 positive stem cells for the regeneration. And interestingly, our team's prior work shows that the CLU protein has the function of self-protection and anti-fibrosis. So we hypothesize that the CLU has the function of improving the condition of the intestine. So we have done our research in 4 parts. First, we have got the CLU gene full knockout mice and conducted a controlled experiments with the wild-type mice and with the no radiation and ionizing radiation, respectively. And the body weight changes and the survival rates were recorded. And from above, we can know that the CLU knockout mice showed a marked weight loss and lower survival rates after radiation. And we have collected their intestinal tissues and measured their length. And you can see that the CLU knockout mice showed a more severely damaged. And we have performed the HE staining on the intestinal tissues. And you can see that there's a decrease in the number of the creeps of the intestinal tissues. And we have also performed the immunohistochemical staining of Ki67. And you can see that there is a lower intestinal creep proliferation ability of the CLU knockout group. And we have also performed the immunofluorescence staining. And the CLU knockout group showed more severe chromosome damage after the radiation. And in order to further verify the effectiveness of the CLU protein, we administer the CLU protein back to the mice and treating them with saline and recombinant human CLU, respectively, and also recorded their body weight changes and the survival rates. And we have collected their intestinal tissues and performed HE and Ki67 immunochemical staining. And all of the results show that the RH-CLU administration can significantly improve the condition of the mice. In order to improve the medicinal efficacy, we decided to use the AlphaFold2 to design and synthesize 21 potentially active peptide segments for the subsequent experiments. And we have also established the human small intestinal organoids for the subsequent screening of the peptides. And now we have successfully completed the synthesis of the PDA nanoparticles that we call the polydopamine nanoparticles and verified their functions. And we have combined our drugs with the PDA nanoparticles and built the delivery system. And the PDA is very important to our project. It has special advantages. First, the PDA nanoparticles can specifically bind to the ROS. ROS is a harmful substance after the radiation injury and can cause a severe inflammation. And ROS can consume and scavenge the ROS, thus inhibiting the inflammation. And what's more, the RS binding can degrade the PDA, thus achieve the controlled release of the drugs. And this is a process how we build a more advanced system. We first load the glue onto the PDA, and then we got the intestinal cell memory, and then we build the targeted delivery system. We call it IM, PDA, glue-tied targeted delivery system. And we have labeled the delivery system with the DIR, and the targeted delivery to the intestinal tissues was observed via the small animal imaging system. And this is our innovation. We are the first to attempt to use the peptide therapy for the radiation injury. And we have made a new PDA nanoparticle-based targeted delivery system and achieved the controlled release of the drugs. And for the significance, we have developed effective and safe radioprotective drugs, and also built a new IM-PDA glue-tied therapy system, and have explored the bioavailability of the drugs. And this is our team's work. We welcome your criticism and suggestion. Thank you.
So, thank you for your excellent presentation. A question about your induced mouse that you induce is like a recessive cruise, right? The cruise mice.
Cruise mice.
Because usually, if you use what the protocol that you use to induce your mice to be recessive in cruise? How do you make a cruise mice?
Cruise mice.
CLU mice.
You say about how to knock out the gene?
Yeah, knock out, right.
We use the CRISPR-Cas9 and to use the full knockout mice. Yes. Yes, full knockout mice, and conducted a control experiment with white mice. Yes, and I give them with saline and becoming cool.
So then, your work is using a nanoparticle with a peptide to recover the what you said that in the intestine, right? For the cruise mice.
You say about this one?
Right.
How we build it?
No, I just question this. What you use a white type or you use just the one that you knock out mice by using this biometric nanodelivery system?
We use the knockout mice to verify the effectiveness. Yes, and in future we plan to give the glue administration to the white type mice too, and to explore the pre-preventive effect of the glue protein too.
So what about the fitness between the wild type and cruise mice? Because usually after you knock out it, usually the knockout mice, the fitness will go not as good as not healthy as the wild type. Did you measure it?
Yes. Now we found that after the knockout, we only find that there is something different after the radiation injury, and the intestine condition is worse. Found that, but we in the prior work we have looked to many literature, and the glue protein has many functions, such as it can cure the Alzheimer's disease, and our team's prior work also shows that and it has the function of the self-protection and anti-fibrosis in the liver, especially.
So it's very interesting to construct this nano delivery system. I want to ask, what is the yield? How many percent that you obtain for each batch of your preparation of this delivery system? Because you use the intestinal cell membrane to be the encapsulate, right? The yield. Yield. I mean the percent yield of this delivery system of one batch of your preparation. Do you know what I mean?
How do I load this or the process? Yes.
Yeah.
We first load the glue onto the PDA and with the peptide binding and hydrogen binding, and we have and then we build the organoids and collected their intestinal cells and collected their cell memory, and by collect and digest with enzymes and centrifugation. And yes.
Okay. So how do you check that it will become this delivery system?
It can bind naturally in in PBS. We find that yes, it has the chemical binding. Yes.
So do you check the encapsulation capacity in the system as well?
Capacity.
Yeah, I mean encapsulate that to make sure that all the peptide and the nanoparticle encapsulate into the intestinal cell membrane. How do you check it?
How do we check it?
Yeah, check.
We have made this small animal imaging system, yes, and we labeled it with DIR, and we can find that the intestinal tissues, can see that here, yes, it is, find it on the image, yes. Thank you.
Can I have one short question? Sorry. How you can, because did you treat the mice with also with the empty PDA capsule with no clue CLU protein inside?
You say about the clue protein?
Yes. So just the capsule, just the empty capsule, no loaded CLU protein.
Yes, they has two functions together. Clue may has the function of regeneration of the intestinal tissues, and the PDA has anti-inflammation function. Yes, they are become stronger when the two strands is together.
Yes, but do you have a control that have no CLU protein in the mice?
The first experiment we conducted only clue proteins treatment. Yes, in the PowerPoint, the first experiment.
Okay, all right, thank you.
Okay, thank you.
Thank you for your excellent presentation. Please give a round of applause.